Neely, are you downstairs? It's like, that looks familiar to me. There might be a study. Well, I don't know if they're open or not. There are some study rooms on the second and third floors, but they often fill up. But there are little rooms that you might be able to, if you get there at the right time, you can have your own little room in this building. All right, so, oops, I hit the wrong button again. All right, so I'm going to go ahead. It's according to my clock, it's time. I want to do an attendance check in a bit, but I don't think that's important just yet. I can do that while you're doing something else. So I'm going to go ahead. I'm going to share this PowerPoint. Let me know if it shows up. Is the PowerPoint visible to you now? Someone give me a thumbs up or yes. All right. Dankeschön. All right. So this week for Bio 2 Lab, we are going to start with Lab 2 in the manual. Now, my hope, COVID permitting, is that next week we will do Lab 1 in person, which will be microscopes and pond water, which I normally like to start the lab with, but I'm hoping it'll all work out where we can do it next week. Okay, so quick question then. If you have the manual and you read through it, there's a pre-quiz at the beginning, which I'm not collecting, obviously. But the first question is, in stats and statistics, what's the difference between little n and big N? Does anybody have an answer for that?
Big N is the whole population and little n is the sample.
Good job. All right. So when we're talking about statistics, which is part of what we're talking about today, is just how do we look at variation in a population? How do we think about it? How do we quantify it? How do we make sense of it? And one of the things we have to think about is, how do we measure it? How do we make a figure of it? How do we talk about it to other people? And in the case of statistics, you'll often see this letter N. If you took Bio 1, you always know, N equals 3. We did that a lot in our figures. If you took Bio 1 here, it's the sample size. How many individuals did you look at, for example? Where that's little n, whereas big N is the entire population, like all the members of the population. Very rarely in biology do we get to look at all members of a population. We have to take a chunk of them and examine them and then infer what the rest of the population is probably like. So I found this online, this image here, there's a whole population, which in reality might be millions of individuals, not, 20, but we take a sample of them and hopefully we get a good representative sample. In this case, this sample is not necessarily great because there's more diversity in the population than in our sample. So we like to have a big sample size, of course, get as many as we can, but in reality, we don't have enough time or enough funding or whatever to sample everyone. So we do a sample. And so the symbol for that is lowercase n. All right. I just wanted to start with that today. All right. So this is Bio 2 Lab. I've never done a lab this way before, like leading people through it. So if it's a little clunky at times, I apologize, but I hope we don't have to do too many more this way. So when it comes to lab, please don't miss more than 2 labs. If you miss more than 2, you can be dropped from the course. Another important thing to note is that you need to do your own work on your own time, on your time. So what I mean by that, and turn it in on time. What I mean by do your own work is, in lab, we work together a lot of times to collect data in a group. But when you go to make a figure, you should do the work yourself. When you go to write a caption, do the work yourself. It's you have to show me that you have the skills to do the work, not that your lab partner can email you a file and then you can turn it in. So do your own work, and sometimes you might help someone. And but if you do help someone in your class, or if you receive help, that help should be instructions on how to do something, not here's mine, why don't you copy it? That gets students in trouble from time to time, so be careful with that. And what happens if you're going to miss a lab? What if you have COVID, or what if you're sick? Get with your lab mates to get the data, and you're still responsible to turn in your assignments on time the following week, etc. And what I'm going to try to do, I'm the first lab of the week. We're all the first lab of the week. If it's on Zoom like today, I'm going to record it. And if it is in person, I'll probably try to record it too. That way, if you miss, you can watch the pre-lab video, and then you can get the data, and you can still do the assignment for that week, rather than have you trying to go to somebody else's lab or something, which could be messy during the time of COVID. We want to avoid that. All right, so we talked about in lecture evolution by natural selection. We talked about some observations and inferences that Darwin made. Today, we are going to focus on the observation Darwin made that individuals in a population vary in their heritable characteristics. So like this population of ladybugs has different color patterns, different spot patterns. Any population of organisms that are not clones of each other will have different alleles, different genes, and they will have variation. And that is absolutely necessary. That's like the foundation of evolution by natural selection. Without variation, evolution is not going to happen. And so I am going to forego this slide. Now, my TAs that are watching, if you're teaching for another instructor, I would go ahead and try and work with this. But all of my students, I believe, we just did this the other day, right? Do all these words look familiar? We just went over these in class. I made students define them. But these are words that we'll have to be able to distinguish in the next couple of labs to understand what's happening. So this week, we're doing this lab. Next week, I plan on doing the microscope lab. And then the following week, we will do a microevolution lab, which will involve genes, alleles, genotypes, phenotypes again. So we'll want to keep these words fresh in our minds. All right, so for natural selection to work, we have to have different genetic traits, different alleles, different variations of a gene. And then, of course, some of them leave more offspring than others, and that leads to adaptation to an environment. We're not going to focus on adaptation today. We're going to focus on how do we quantify, how do we make sense of this variation in a population. Now, you can use the same tools we're learning today to look at other kinds of variation as well, like ecological variation and things, but we're going to focus on the idea of variation due to genetic factors. So when we look at a population, we can see that some variation absolutely has to do with genetic factors, like this individual here. This is congenital generalized hypertrichosis. All right. Does anyone know what congenital means? What does that word mean when you see it on like a disease or something?
Does that mean having like both reproductive parts?
That's a good translation, but it's actually not what this one means. You're on the right track, but not in this. Genital does have to do with something to do with genitals, but anyone else heard this word?
Does it have to do with like before you're born? Like, because I
Okay.
know there's a congenital heart defect.
Right, a congenital heart defect. Congenital means from the moment you came from the genitals, basically, the moment from birth. Okay, when the moment since you were born, you had this problem. Okay, it's not something you developed later. That's what congenital means. You developed it later on. You developed it from the time you were born, basically in the womb and after. So congenital generalized hypertrichosis means this is something this individual was born with. It's not something. It's not like they lived near a nuclear facility and they drank poisoned water and they developed this later or whatever, right? This is something they were born with. It's a genetic trait, like we talked about. They have an allele or two alleles. In this case, it's an allele found on the X chromosome. It's an X-linked gene that runs in some families. Generalized hypertrichosis generalized means all over the place, and hyper means overactive hair follicles, basically. So this is clearly a genetic trait. Now, in reality, a lot of our traits, of course, have to do with our genes, the genes we inherit, but also the environment influences us. And it's obviously very complicated because in reality, we have genes and the environment, and both are factors in how we develop. Can anyone think of how the environment might influence somebody in your development? And by environment, I mean anything outside of yourself.
Like access to food and water. I mean,
Yeah, sure.
like just
Access
basic
to
access.
nutritious food, access to healthcare, diseases. Those are all how much sunlight there is outside. If you're living in the at the equator, it's going to be a different environment than if you're at one of the near one of the poles. Those are going to be all environmental influences that can affect your development. It's not going to change your genes, but it might change some aspects of your development. Now, I'll give you an example, a concrete example in a second. So bear in mind that we have lots of variation in the human population. For example, we might have, here we go. Here's a question for you. Big H, big H. Big H, big H. Which one of these things is big H, big H?
Genes.
If we write it out, big H, big H.
Oh, of those things.
An allele.
The genotype.
I heard almost every answer on the screen. Okay. So allele is H, and you have two copies of big H. What is that then?
That's the genotype.
That's the genotype. Genotype is which two do you have? Because remember, this person is diploid, 2N. They have two copies of every chromosome, so they have two copies of every gene, and in this case, they have two copies of the same allele of that gene. So the allele in this case is big H. The gene is for earlobes. The allele is big H. The genotype big H, and then the phenotype is the expression of that. So you could be big H, or you could be big H little h and have the same phenotype, detached earlobes, where your earlobe hangs down a little bit. And if you have little h, that's a genotype. That's homozygous recessive. Then you have attached earlobes, where the earlobe kind of flows right into the side of your head there. It's not dangling free, right? So that's not probably one that's going to result in natural selection affecting somebody, but that is an example of genetic variation in a population that results in slightly different developmental features, something that we can easily see. So that's just one example of how you might be a little bit different than the person next to you on your Zoom screen here. But of course, as humans, we have tons of variation, right? We have all kinds of variation, not just earlobes, but physical traits that we can see, eye color, hair color, skin color, all of that, but also which kinds of enzymes we have. I use the example in class. Some of us are lactose intolerant. Some of us can digest lactose because we have different enzymes that are on or off, depending on what genes we have. And that's just one example of many. Here's an example I found online that I thought kind of drove this example home. These are identical twins, but they were separated as children and raised in different environments. And which one do you think probably had more nutrition, better food, more access to good, healthy food?
The one on the right.
Probably the one on the right. So these two individuals have the same genes, the genes that code for how tall they can get. But that doesn't mean you're going to be that tall, because it also depends on the environment, how much nutrition you had while you're growing, if you caught a horrible disease while you were growing, it might keep you from getting that tall, etc. So the environment and the genes both interact to lead to the phenotypes that we see. Now, where does this variation come from? We've talked about this briefly in class. In Bio 1, you may have learned about mutation, where DNA copying itself can have errors that can lead to brand new alleles, or one allele turning into another allele. This doesn't happen in someone who's already born. You're not going to see this passed on. I mean, you can have a mutation like in your little finger or something, but this only gets passed on if this mutation is in a gamete, in a sperm or an egg. If it's in a gamete, then it gets passed on to every cell of the offspring that that gamete leads to. But if you have a mutation in your little finger, it's not going to be passed on. Now, also during meiosis, independent assortment of chromosomes leads to shuffling of genes around, shuffling of alleles around. Crossing over does the same thing. And then the random nature of fertilization, where you have, you know, a lot of times anyway, one egg and, you know, several million sperm, every one of which is a little bit different from one another. So there's lots of possibilities there as well because of that. So mutation, though, is the one that creates new traits. The other examples are more like shuffling a deck of cards. You're not going to change the colors of the cards by shuffling them. You're just going to get different combinations. Just like all of these different processes here shuffle around pre-existing alleles and provide them in different combinations when gametes are produced or when fertilization takes place. But this leads to variation. That means that you and if you have a sister or a brother, even as long as they're not your identical sibling, they are going to have genetic differences from you, even though you share the same set of parents. They will have slightly different genetic makeup, different alleles because of these processes. All right, so that gets us to the stuff that makes students go, ah, when they look at it at first. Now, if you have your manual, the manual starts out with talking about DNA transcription translation to RNA. That's kind of the same thing as saying genotype to phenotype. And then it goes into the stuff I'm about to talk about. Now, I will point out that the figures I have on the PowerPoint are from a different data set than is in the manual, but the same concept is here. And TAs, those of you, by the way, there are teaching assistants watching this as well in the class. That's why I refer to the TAs. From time to time, they're watching so they can be up to speed when they teach later this week. If I get the time, TAs, I wouldn't mind updating this PowerPoint to have the exact same data as in the manual. If I get a chance, I'll try to do that, and I'll let you know if I upload that file. Okay, so in our example, we go to an island and we're measuring bird beaks. Why bird beaks? Well, it's a very Darwin thing to do, so we'll use that as an example, right? So you're on an island with lots of bird beaks. And this graph here, does anyone know what kind of graph this is, what the name of this kind of graph is, where you have frequency on one axis and then you have beak length on this axis? What is this called? Where you see the distribution of a trait. It's on the screen.
A histogram.
A histogram. Good job. Thank you. All right. When you draw these things, ideally, you know, without looking at individual columns, they're often drawn, and you hear the word bell curve sometimes. Yeah, you know, in a normal distribution, you have a bell-shaped, as they call it, distribution. My drawing's not very good there, but what we have here on this axis, on the x-axis, is how many birds were there with a beak that long, and then the y-axis is how long are the beaks, right? And so you can see in the example in your book, I have a simpler data set. I don't know if it'll show up because I have a fancy background on my camera right now, but you have this data set here, a little bit simpler, right? In your manual, but on here we have a wider variety. This symbol, by the way, this X with a line over it, stands for average or mean. That's what that symbol stands for. So in this example on the screen, which is a little bit different than your manual, we have quite a bit of distribution, but we do have an average. The average on the one on the PowerPoint is 18.8 millimeters. That's how long the beaks are on this example. And so if I put that on this histogram, that's about right here. And you can see there's variation around that. Now, what kind of data might I, as a professor, look at during this class that relates to you and something you might care a lot about? How might I use a graph like this that has nothing to do with evolution? Let's say after you take a test.
Yeah, grades.
I often look at I often look at grades on this kind of a graph, because I can say, okay, the average was here. Now, are there a big pile of F's or a big pile of A's, or are they equally distributed? What happened on this test? How are grades distributed around that average? I still hear talking, but it's breaking up. It sounds like we're communicating back in time. I'm going to scroll through here and say hi. Julia, can you hear me? I'm going to wave. Hi. Marcus, how you doing? Emily? Mackenzie? Ty? Is it Allie Dixon? Allie? I see Neely. All right, now help me out. Is it I have to, is it just Jasalyn? How do I say your name properly so I don't embarrass you?
It's Jesselyn.
Jesselyn, thank you.
I go by Jessie though.
Jessie? Okay, Jessie. I'll try to remember that. I'll get it by the end of the semester, right?
Okay.
And Tanner, how you doing today?
Doing great.
Tanner's one of our TAs. And we have a few other TAs in here. I think Jackson's in here and Kira's in here. And yeah, how you doing? And there are more students I haven't said hi to. I'm just winging this here. So if I don't see you, I didn't call on you yet. All right. So this is our distribution of bird beaks. And we can see that there is an average, but not, what would this graph look like? Let me just ask you this. What would this graph look like if all the birds had the exact same beak length? What would it look like?
Would it just be like one, there'd just be like one long line?
Yeah, there'd just be one column in the middle and that's it. That's what a population with no diversity would look like in this case, right? With this kind of graph. But here we can see there's a lot of variation. There are birds with bigger beaks, there are birds with smaller beaks, and there are birds with somewhat average beaks. So if we draw the shape of this, it does come out to be somewhat of a bell curve, as we call it, that hill shape where there's more close to the average and there's fewer out here along the edges, right? Okay, so we can see that. Now, I want to point out then there's a way one of the ways, there's lots of ways. If you're taking stats, you're going to learn more detail. Okay. We're going to learn just enough stats to be dangerous here. By the way, TAs, Kira, Jackson, et al. Did you have biometry recently or a while ago or not yet?
I had it last semester.
Sweet.
Last semester.
So you all can like rock this lecture and fix it for me when I screw it all up.
I had it last year. Of course.
Last year. Okay. Hi, Zach. Hey, you got 2 for 1 on that screen. 2 TAs for the price of 1. All right. So here we have a statistic known as standard deviation. Standard deviation is a number that kind of gives you an idea of how spread out the data are. That's how we're going to use it. And if you calculate this in Excel, for example, you're going to get a number. In this case, I calculated it and I got 4.4. Now in your manual, I have a slightly different value because it's a different data set. But in this example, we have 4.4. So what does that mean? How in the world is that useful? Well, with standard deviation, if you find the average and then you draw a line 4.4 millimeters to each side of that average, then all the birds within that box constitute around 67 or 68% of your data points. It gives you an idea where about almost 70% of your data will be between those two lines. And then if you add another 4.4 to that and go even further out, that's going to be 95% of your data points. And if you add another 4.4 to that, that's going to be at least 99% of your data points. So the point is, a lot of times we don't even show histograms. We don't graph histograms when we're talking about data. We might show a column graph and just throw out how many standard deviations wide the data were. or I should say, we'll tell you what one standard deviation was. And then you got an idea of how close these numbers were to the average. So if you had a data that was all really close to the average, you'd have a small standard deviation. Whereas if you have data that's really wide and goes out for a ways past the mean, then you have a larger standard deviation. So this tells us how spread out, how much does the how much does the data vary around that average or mean? And so there's an example of that in your manual. One of the things I asked to do in the manual is in the manual example, the standard deviation is 1.1 millimeters. So I said, draw a line on that figure on page 13, 1.1 millimeters on each side of the average, so you could see what one standard deviation would look like on that data. And I'm not going to collect that, but that's something you can do in the manual to kind of visualize this. Okay, now the other way we can depict the data, which is the way we typically do in class, is rather than have frequency on one side and different bird beak lengths on the other side, is we average all the lengths together. We just get an average of all of those lengths. And then we can stick an error bar on it. In this case, this is standard deviation. So this is 4.4 millimeters on either side of that average. So from here down to here. And so, yeah, this data looks way different than the past graph, but it's showing us the same basic thing. It shows us what the average was, and it shows us one standard deviation above. So you could say this is plus one standard deviation, and down here is minus one standard deviation. So you get an idea. You know that 68% of the data points used to calculate this average fell between there and there. Okay, that gives you an idea of how spread out things were. Now, we're probably not going to make very many, if any, graphs in Bio 2 where we show a standard deviation bar like this, but it is possible. I just wanted to show you what that would look like. You can do it. Now, this would be if you would do this if you're describing a population. Like population 1 had an average beak length of 18 millimeters, and this is how much it varied around that average. That would be describing a population. But a lot of the times in Bio 2, we're not just describing something, we're comparing things. And when we compare things, we tend to use a different statistic, one that you're already familiar with if you took Bio 1 here, but we'll get there in just a little bit, okay? All right, so I am going to, first let me have a look at this slide. How do we calculate averages? How do we calculate standard deviation? I want to do an example. So in Excel, the formula for average is you type in an equal sign, you type the word average, and then you use your mouse to select the data. You literally highlight it with your mouse and you put that within two parentheses like you see here, hit enter, and you get the average. How do we calculate standard deviation? Well, we're going to do this in Excel rather than by hand. I hope that doesn't make you sad. We're going to type equals stdiva and select the same data that you select for your average and then hit enter. Okay, so here's our question. What is the average and standard deviation of the height of the sample of Bio 1441 students? And I also ask, what is the sample size? Well, we'll get to that in just a second. All right, so what I want to do, I'm going to open a chat window and I'm going to try to send you all a file. Let's see if it works. If not, I'll email it to you. I tried to make it easy for us today. Oh, and of course it didn't work. Try again. Try again. Try again. Oh, and it hates me. Okay. All right. Well, I'm going to email it to you. And I'm going to check my email because I think I have some emails from students who had trouble getting into the lab or something. So give me a minute, but I'm going to email you this file. Give me a second. It's not wanting to send through Zoom, so I'm going to send it to you via email. All right. Monday lab. Email. I'm going to call this data for lab 1. Attaching the file. Lab 2. I call this lab 2 practice data since it's lab 2 in the manual. All right, I'm sending it to you right. Oh, let me put a subject line or data below. All right, sending now. Okay, I've sent you the data. That way you don't have to type it all into Excel. I want you to open that data and I want you to try to calculate the average and the standard deviation for that data. And in order to help you, or in order to make this, some of you may have never done this before, but I bet a lot of you have. It may not be terribly difficult for some of you. I'm going to hit pause.
Come on. It's your turn.
Is this what you wanted?
Yeah. So, okay. So how did you calculate? Would you go ahead and just calculate it again and show us how you did it? So how do we calculate? Let's start with the average. How do we calculate the average?
All right.
So click on a cell.
In that. Oops. Mine pulls it up for me.
Okay. So you can just click it. Okay. So you start to type it, then you clicked it. So average. Maybe. Doesn't want to work when you're showing it off though, right? All right.
No, it's not doing it.
That's okay. Then you just open a parenthesis, highlight the data, close the parenthesis and hit enter. There you go. There's the average. So the average height in centimeters. Now, how do we calculate the variation around that average, the standard deviation? How would we do that?
So that equals. No, it's not working. And
Hey, let's go outside. Come on.
Hannah, you're unmuted.
Sorry, I'm taking my dog out.
Tell them they're a good boy or girl and give them a pat for me.
She's not being a good girl. That's the problem. She's kind of being a B word this morning.
Well, that's her job. All right. So we have same number. Did anyone get anything other than this? Is this what we got? I can only see a handful of you at the moment, but there is a chat window and you can also tell me. I'll say hi in the chat window to everyone. Say hi, reply to me in the chat hi, so everyone knows where it is, just to be sure. Thank you. Groovy. All right. All right. Well, good job. So that's how we calculate those two values. Now, what I would do, one thing you could do to when you're graphing like that is to also then label which one's which. So right above your average, type the word like mean or average, and right above the standard deviation, type SD, for example. So you remember what things are. That's, something I often do, so I don't forget what I am showing there. Okay, so I'm going to go back then to the PowerPoint. Maybe. All right, 3, 2, 1. Are we good? We got it. It's working. All is good. Okay. All right. So In your book, we have another example then where we're going to compare two populations. And once again, the data on my PowerPoint is different than the data in the manual, but it's the same concept. So we have population A that we already looked at. And let's say there's population B. Now, population A, the birds eat insects. There's a lot of insects on the island. Population B, we have a lot of seeds on the island, and the birds are eating those seeds. But we think population A and B recently split off from one another. So we might wonder, okay, maybe the birds in population B are adapting to a seed-based diet and showing evidence of this adaptation. So we could write a research hypothesis. By a research hypothesis, that's what we did in Bio 1 when we write a hypothesis, right? If then. So the point of this slide is to show you that a hypothesis is an explanation, not just a prediction. So if this were just a prediction, I would say the birds on one island will have a bigger beak than the other island or whatever. But I have an explanation here. If the birds in population B are adapting to a seed-based diet, that's what I think is happening. They're adapting. Then the two populations will have a significant difference in beak length. So that's my hypothesis. Now, the example in the manual on the bottom of page 13 has a second part right here that says "and," which kind of describes how I'm going to tell if they have different size beaks. We don't have to include that. That makes the hypothesis chunkier and a little bit, but you can include that. Like the example in the manual says, if birds in population B are adapting to a seed-based diet and beak lengths from population A and B are measured, then the two populations will have a significantly different beak length. You can put that "and" part in, but you don't have to. But the important thing is here, we think they will have a difference. Now, I did not predict in this case how they would be different. That's going to be important later. How you word this is important. If you predict difference, you're going to do a different statistical test than if you predict which one will be bigger or smaller. So I'll come back to that later. All right, so here's a graph then, a column graph. Now, this is a lot like the column graph I showed you before, except now I'm not using standard deviation. When we compare two populations, we're going to use standard error, or SE is the abbreviation for that, standard error of the mean. And the way you calculate that is you divide your standard deviation that you just calculated a little bit ago by the square root of the number of samples. Okay. So the true population mean is going to fall somewhere between the error bars when it comes to standard error. That's the idea. If you have standard error bars, you're saying, okay, there's variation in the population, and I have these error bars here. Probably if we sampled the entire population, I'm pretty confident that the true average is somewhere between these two bars. And so if the bars are really big, it means you're less confident where the true average is. If the error bars are really tiny, it means you're pretty darn confident where the true average is. Now, in this example, standard error, let's have a look here. So in this example, how could you make, if you're in charge of designing your own experiment based on this formula, what could you do to make your error bars smaller, to be more confident that your data, your samples represent the population? What could you do to be more confident about that? Which side of that equation do you have control over?
How many samples you have?
How many samples do you have? Yeah, you can't control biological variation, but you can control how many samples you take. So obviously the more samples you take, the more confident you are that your average is close to the real average of the whole population. So remember, this is we're using a small n, a sample here, but we're trying to estimate what the whole population has. And the more samples we have, the more confident we are that we are close to that true mean. And that's what standard error of the mean tells us. That we're pretty confident that the true mean of the entire population is somewhere between these two error bars. So we often put those on graphs like this when we're comparing two different populations. And one thing you can do, it does give you an idea of how variable data are around the average a little bit, of course, but it also allows you to quickly eyeball the difference between two samples. If the standard error bars overlap like they do here, we probably can conclude that the two samples are likely not significantly different. I'm not going to say they are or they are not using error bars, but I will say they may or may not be. We'll have to do further tests to say they are or are not significantly different. But in this case, I would say these two samples are likely not significantly different. Their averages are a little bit different, but their error bars overlap along this axis here. So that means some of the variation within the groups overlaps with one another quite a bit. So probably they're pretty similar in beak length. Now, if you have a sample like this where the averages are different and the error bars do not overlap, like we see here, then we could say the samples may be different. They may differ. We can't say that they are significantly different, because what I want to point out here is the word significant has a very specific meaning in science. That means you've done tests to show significance, like a t-test or an ANOVA analysis. Has anyone done a t-test before? I guess I can only see like 5 of you at once right now, so I can't tell if anyone has their hand up. If you have done a t-test before, say yes in the chat. I'm not getting anything in the chat, so I'm going to assume that's a no. Okay, that's fine. That's why we're here. Okay. All right, so let's just do an example of a different kind of graph. This is a scatter plot, or well, it was made with a scatter plot feature in Excel. It's a, like a line graph essentially, where we have averages here. These are data points. We have fertilizer concentration down here and how much plants grew over here. So this one may not really be genetic data or, variation due to evolution, but due to fertilizer in this case. How would you interpret this graph? If you just had to put into a sentence, what happened as you added fertilizer? What did it do to plant growth? I'll call on Emily since no one's speaking up. Hi, Emily. What did fertilizer do to plant growth?
The more fertilizer you added, the more the plant grew.
There you go. Pretty simple, right? Now, what if I had error bars on this data and it looked like that? Is that going to change our interpretation? Anyone other than Emily want to pipe in on that one? I'll make Jackson pick somebody. Is this data point, do you think it's likely significantly different from this data point? Why not? I see Abby shaking her head no. Why not?
Because the error bars overlap.
They overlap. But is this data point likely different from this data point? Probably, because data point 1 and data point, whatever this is, 5 or whatever, their error bars do not overlap. So these show a difference in the average. I'll draw a line connecting the averages. We can still see that trend. The trend is as you add more fertilizer, growth goes up, but it may not go up significantly between each sample. But some of the samples are different, likely different from one another. Of course, we can't say they are significant or are not significantly different yet. This data would require further statistical tests, in this case, like an ANOVA analysis or something, which we're not going to do today. All right, so how do we calculate standard error? You select standard deviation. So literally you calculate it like you did before, and then you basically put an equal sign, and then you click on the standard deviation and you divide it by the square root of the sample size. And the way you do that is, actually, I should probably put a parenthesis here as well. You divide it by SQRT, that square root of the number of samples. So for population 1, we're getting an average of all these numbers, right? So there was 1, 2, 3, 4, 5 samples in that population. So N is 5. And same for population 2. So for both of these populations, the sample size is 5. So what I want you to do now is calculate the average of sample 1 and sample 2, which you've already done sample 1. It's the same sample. And then I want you to calculate the standard deviation for those 2 samples. All right. So, and if you want to chat about it, I'm going to try to stick a TA into the different rooms. You have the data. I sent you the data already. If you don't know how to get to the data, let me show you real quick. Let me figure out what the heck I'm doing. Okay, so let me share my screen again. Do you see my data or not right now? Did I share correctly? No?
No.
Okay. Okay. Let me try again. How about now? Okay. So on your Excel sheet, you already looked at this one. If you go down to the bottom, there are 3 different pages. If you go to page 2, it'll have this data on page 2, and you can calculate averages. You'll have to calculate standard deviations, and then I want you to convert that into standard error. And if you know how, go ahead and make a column graph and stick error bars on it. If you know how to do that, I'll know how much I need to teach it on the next slide. But I bet we'll need to cover that for a lot of us. But go ahead and try it if you know how. Meanwhile, I'm going to stop the share. I'm going to break you out again so you can talk to one another and help each other out if you're stuck. Hannah Grace Fritz Johnson. Okay. Boom. Gotcha. Smith. Gotcha. Richardson. Trey. Gotcha. Dixon. Schoffer. Jaw. Okay.
One here.
All right. So I haven't seen how far you've all gotten. And I know some of you have not had bio one or like, I don't even know what's going on. And others are like, that was easy. So we have I want to point something out real quick. Am I sharing my whole screen? Are you seeing like my email right now, for example? Okay.
Yeah.
All right. So if you run into trouble later and you want to go to, actually, you can probably just Google biology 1441. And the first thing that probably will pop up is the lab website. And up here in graphing, I have videos on how to do all the stuff we're talking about today. If you need a refresher after this lab's over, when you're working on homework, if you get stuck or something, I have videos up there. I also have a few other videos on a couple of things from lab. There's the schedule of when labs meet. And also there are pre-lab videos that I often post up here that could help you be ready for a quiz or something. So that's up there. And then in the room schedule, if you forget when your lab is or who your instructor is, there's a schedule up there that has the labs of the week laid out for Bio 2. So that's all on the lab website. There's also a link to it in Blackboard if you're in, those of you in my class. So you can find it that way too. Okay, so here we are. I'm going to go ahead and walk through this one to make sure. Let me, first of all, you see my Excel sheet? Are we good there before I start talking? Okay. All right. I'm going to zoom in on it a little bit. Okay. So if I was going to compare this information, first of all, I would go down here and I would go ahead and type sample 1 and sample 2 right here. So I remember what's what. And then I'm going to label over here. This is going to be average. This is going to be standard deviation and my standard error. Those are the things I need to calculate. So I like to label it the same way every time, so I remember what I'm doing and what's what. So if I open this file again in 2 years, I'll still know what it means. So to calculate average, I type an equal sign. What do I type next? I know Marcus knows. What do I type, Marcus? Oh, you're still muted, but that's okay. I think I saw your lips say the word average.
Yeah.
Average, then you open a parenthesis and I highlight all these numbers from sample 1 that I want to get the average for. I close my parentheses and I hit enter. For sample 2, I can do the same thing, average. I highlight these numbers from sample 2 and I hit enter. And now I know the average height of students in sample 1 and I know the average height of students from sample 2. Now I want to calculate the standard deviation. To do that, I put an equal sign and I type in ST_DIVA. And I open a parenthesis again. That's how you do formulas in Excel. And then I highlight the exact same numbers that I use for my average. And I hit enter. Do it for sample 2, equals ST_DIVA, open a parenthesis, highlight the data from sample 2, and I hit enter. Now I have the standard deviation for both samples. And I have the average, of course. Now I need to calculate standard error. Now, the reason we have to calculate standard deviation is that Excel doesn't have an automatic standard error calculator when we're doing it this way. So to calculate standard error, we put an equal sign. And then I literally just click this standard deviation because I'm going to divide standard deviation. I put a divide by sign, a slash, and I have to divide by the square root of the sample size. So the sample size is 5. The way you know what your sample size is, it's the number of people, in this case, the number of numbers you added up to get your average. Okay, that's your sample size. And how many did I add up here to get this average? I added up 5. Okay, so divided by the square root of 5. Enter. And that's my standard error. Now over here, I go equals, and I'm going to click standard deviation divided by the square root. Once again, it's the same sample size for both of 5. And so that is how we calculate those. And there, if you forget what the formula is, it's on the top of page 14. The standard deviation divided by the square root is sample sizes. So there we go. Now, if I was going to turn this into a column graph, first thing I would do is highlight sample 1 and 2, the 2 words, and I would highlight the 2 averages. So if you haven't done it, follow along with me here. Highlight the 2, and then go to insert. And then you're going to put, and of course, if you have a Mac, things will be in a slightly different place than if you have a PC. I have a PC. If you have a Mac and my videos are all on PC, I do them all on PCs. Bear in mind that all the computer labs on campus have PCs and will work just like my videos. But the Macs aren't that different. Now, if you go to insert, you can then pick what kind of graph you want. And in data like today, where you have 2 categories, sample 1 and 2. So you have categories on one axis and numbers on another axis. That's going to be a column graph. So I'm going to click on column graphs, and I'm just going to pick this first one here. There it is. Boom, column graph. A few things that I'm going to recommend you do. First of all, delete the title and delete these lines, these annoying lines that are really unnecessary. It cleans up the graph quite a bit. Okay, now to show you what we would do next, depending if you have a PC or Mac, but somewhere there should be, when you click on the graph, there should be a plus symbol, and or I know sometimes on a Mac you have to go over here to like add chart element or something like that, depending on what version of Excel you have. But things that I need to do, what does this graph need yet? Those of you who've had Bio 1, what am I missing?
Error bars.
Error bars is one thing. So let's do that first. I'll click the chart element symbol. I'm going to go down to error bars. And if I click it, I might say, yay, I'm done. But it just stuck random error bars on there. I don't know why it does that. I don't know what those error bars mean. Those are not right. I need to put these numbers into my error bars, that 3.2 and that 3.3 value down here. So I have to go to error bars. When I go to error bars, I have to click this little, I'm going to call it a chevron, the triangle thing right there. And I go to more options. I'm going to leave this screen up for a second. So if you're doing it and following along and doing it too, you can do that and toggle back and forth. So you click more options. And then at the very bottom of the screen, you go to custom. Now, just to warn you, sometimes this screen doesn't come up and you'll see something like this. And it's like, what color do you want your error bars to be? Well, that's because this bucket is selected. You need to select the graph over here on this menu. That frustrated me at first when I couldn't find this option. So I go to specify value. Okay, it says positive value and negative value. Now, all you got to do is click on this little arrow next to positive value and highlight these 2 numbers that you calculated, the standard errors. And then you do the same thing for the negative value, the exact same 2 numbers. And then you click OK, and now your error bars are correct. What else does this graph need?
Axis titles.
Yeah, we need to label our axes. So in order to do that, we click on the plus button and we go to axis titles, and I just click it, and now I can label it. So I'll say, I'll just give it some kind of title down here, 1441 section. and average height. And I need a unit. You always need units. And I'm going to open parentheses and say centimeters. Oops. And so now I know what's going on this graph because I labeled my axes. So to do that, once again, I went to the chart elements either here or up here in the corner, and I clicked it, and I went to axis titles. And when I checked it, it stuck titles in that I just had to fill out. So now we have our graph made, and I can't walk around. If I were in class, I'd walk around and have you show me all your graphs. I'm worried that would take forever trying to get everyone to screen share to show me your graph. So I might check on a lot of you here on the next sample though. So what I want to show you next, I'm going to go back to the PowerPoint, I hope. Is the PowerPoint visible now, or is it still the Excel sheet?
PowerPoint.
Okay, good. Thank you. Okay. All right. Now, well, if we look at that data, let me open that data back up again for just a second. If we look at this data, I would say that data may be significantly different. Wouldn't you say? The error bars don't overlap, right? This error bar here and this error bar here don't overlap in space. If you look at the y-axis, they don't overlap over the same numbers. So we can't say they are different, but we can probably say they may well be different, but we have to double check. You know, maybe we have a bunch of tall folks in sample one. You know, maybe we have the basketball team in that section and a normal population sample two or whatever. You know, I don't know. This is just sample data. So in order to tell if the samples are different or not, significantly different, we can do a t-test. And I have information on this on page 15. And so I want to walk us through this for just a moment. So I don't want to lose you here. This isn't really that hard, but I want to explain it a little bit and talk about statistical hypotheses for just a second. So first of all, this is the formula for a t-test in Excel. If you want to write it down or something, you can. Equals t-test. You don't have to write down the words array, or whatever. Excel will tell you that. But the formula is equals t-test, and then you open parentheses, okay? Now, t-test will work when you have like a column graph with 2 columns. If you have 3 columns, 4 or 5 columns, a t-test isn't what we're going to use. And in fact, most of our data will not be that complicated in Bio 2. You'd have to do something else, like an ANOVA analysis or something. We're going to stick to simple t-tests for now, okay? And when it comes to tails, that word tails here, that's asking you, you know, you're looking at 2 different populations. If we draw another histogram, it's asking, do you want to compare just one tail or both tails to each other? Essentially, this is a crude explanation, but ultimately what we're asking there is, do you want to look for a difference? Is there a difference between the 2 samples, or are you predicting which one is bigger, like which one has a longer tail on this side, let's say? What I mean by that is, if you're doing a one tail test, you're only looking at one side of that distribution. Like I predict population A has a taller population than population B. That would be a one tail test. If you're just saying, I predict there's a difference, I don't know if one's taller or one's shorter, I don't know which way is which, you just do a 2-tail test. And it's written out on page 15. A 1-tail test is when you predict a direction of difference. And a 2-tail test is when you are just looking for the existence of a difference. And so we're going to just do a 2-tail test for now. And that's probably what we'll do most of the semester, or a lot of the semester, is a 2-tail test. And then when it comes to type of t-test, for a lot of our t-tests, we're going to choose the number 3 for that one. And I'll explain that a little bit more later, but just know that we're going to typically do number 3. And I'll show you an example of this in a second to make this make more sense. Okay, now we have a biological hypothesis like for our bird data before. We predicted that there would be a difference in bird beak lengths from those two different bird populations. Now, that's a biological hypothesis or a research hypothesis. In statistics, when you do stats like a t-test, you also have what we call statistical hypotheses. And that's basically what you're testing with the t-test. And first things first is you have what they call a null hypothesis, which is the symbol for that is H sub 0, H0. That means, so H sub 0 is the null hypothesis. That is just saying, okay, there is no difference. Null means nothing. There is no difference in this, in bird beak length between the two populations, or in our case, there is no significant difference in height between the two populations. And then you can have an alternative hypothesis, which is H1. There is a significant difference in height between population A and B, or 1 and 2, or whatever. If you said if H1 is there is a difference, but you're not predicting which one is taller or which one is shorter, that's a two-tailed test. If H1 said population 1 is on average taller than population 2, you're predicting direction there, then you would do a one-tailed test. Now let me show you how to do this and how to interpret it. Carafit quick and messy style here. So here we are on this data, and I want you to follow along with me if you can. I know a lot of you don't have two screens, so I'll go slowly here. I'm going to do a t-test right here. We start off with an equal sign. And there are different ways to do t-tests. Let me be clear. I'm doing it this way because it works this way on Macs and PCs the exact same way. And I think it works in Google as well, on Google Sheets this way too. You type an equal sign and t-test. And then I open a parenthesis. What I want you to see, if you can see my screen for a second, is that it gives me options. It says array 1, array 2. And notice there's a comma between each of those things. Array 1, comma, array 2, comma, tails, comma, type. All right, so that's telling me what to do. Array just means the columns of data. Array 1 is sample 1, array 2 is sample 2. Okay, so what that means is I highlight sample 1, and then I have to put a comma to let it know, okay, that's sample 1. That's, population 1. Or whatever. And then sample 2, I highlight that data. And then I put another comma. And then it says, how many tails? Well, it says 1 or 2. I can literally type the number 1 or 2, or I can click it, as you can see on my screen. I'm going to do two-tail distribution, comma. And then it asks me for the last set of data. And I'm going to choose number 3, 2 samples, assuming unequal variance. That's just a safe bet. I'm not going to explain that exactly, but in class, when you're designing your own experiments, you're either going to probably choose number 1 or number 3. And normally it'll be number 3. The only time you choose number 1, which if you can see it on my screen, I can't tell what you can see exactly. 1 is a paired t-test. And the only time we do a paired t-test is if, let's say we're testing heart rate, and you're testing the same individuals before and after a treatment. Like I'm testing everyone in the class's heart rate before exercise and everyone in the class's heartbeat after exercise, but it's the same individuals. That's a paired test. But that's not what we're doing here. We're looking at 2 different classrooms. So we're going to choose number 3, and I close my parentheses. So I'll leave that up for a second. You can see I highlighted sample 1, I highlighted sample 2, then I put a comma after each of those. And then I click 2 for a two-tailed test, or I typed in the number 2 for a two-tailed test. And then I type number 3 for the type of test. That's assuming unequal variance, because I don't know if the variance is equal or unequal. So I'm going to assume it's unequal to be safe. And I hit enter. And I get a number. This is our p-value. And the way this works into statistics is if the p-value is less than 0.05. Now, let me ask you this. Is my number less than 0.05? or greater than 0.05? Which one is it?
Less
It's less than by,
than.
it's got a whole other zero in there, right? And so in the 100th position. So it's less than 0.05. That means we reject the null hypothesis, that statistical hypothesis. So if you remember, the null hypothesis was there is no difference between the two samples. Now, if I reject that, that means there is a difference between the two samples. Now, if my p-value was 0.05 or greater, then I would have to accept the null hypothesis. So if it was greater than 0.05, I would accept. And there may be, I don't know. I have to ask, hey, TAs, if you're right on 0.05, what did Reed tell you to do? Accept or reject, or did he not say?
So reject them all.
Reject if you're right at it? Okay.
You got to err on the side of caution.
Sure. Especially when you're like giving people drugs and seeing if they die or not or something. It's always good to err on the side of caution when it comes to statistics. Yeah. Especially in the medical and science fields. Okay. So none of that is intuitive. I understand, but I'm going to have you work through it again on a sample in just a minute. And I'm going to put you in breakout rooms, and I want the TAs also to help. You can screen share in the breakout rooms. You can say, here's where I am. I'm stuck, help, and that sort of thing. And we can walk you through it. Someone says, are we supposed, oh, am I not sharing my screen right now? You just see my face? When did, are you seeing my screen? Okay. So apparently what,
I'm still seeing the screen.
I can see your screen.
okay. All right. Apparently someone was having trouble. Okay. All right. So I am going to show you, let me go. Okay. Go to the next thing here. So once again, that's, and by the way, there's more information on this stuff. If you don't have it memorized from my quick spiel on page 15, I have an example null hypothesis for the bird beak data. I tell you when to use what kind of test, and I tell you how to interpret a p-value there. Okay. So what I want to do then is we go to part 1, analyzing data. And by the way, I do have a slide here on t-tests, which kind of covers what I just said. We will often use a two-tailed test, assuming unequal variance, which is what we did. And once again, I have how to interpret a p-value here. This video, by the way, I'm going to post this whole video and on the lab website for people who are sick this week, so they can watch this very exciting video at their leisure. Okay, so Dr. Gifford gave us some real life data that will appear on page 16 of your lab manual. And this is data from prairie lizards. What he did is he collected lizard eggs in Arkansas, and he collected eggs from these lizards in Missouri. And he took them back to UCA, and he put them all in an incubator at the same temperature. And he measured how long those eggs take to hatch to see if there is a genetic difference in how long lizards from those two different populations take to develop until they hatch. Okay. And so you can see that data here. You can see the Arkansas data, you can see the Missouri data, and you might be able to get an intuitive sense as to if one of them has a higher or lower average than the other. But we want to kind of think about, we want to be able to quantify it and show it and put a t-test on it. So if you look at your manual, it says number 1, what factors might cause variation in the incubation time? Let's just focus on between these two populations. Why would lizards in Arkansas maybe have a different incubation time? That means from the time the egg is laid until the egg hatches. Why might they have a different incubation time in Arkansas versus Missouri?
It's warmer in Arkansas.
Okay, so Missouri is higher in latitude, and it could experience more frequent cold climate. So that's a good one. That's That's the environment affecting or selecting for a certain incubation time. But we can be pretty sure, though, that this data is not due to actual weather patterns, because we incubated them at the same temperature in a lab. But those lizards that laid those eggs are adapted to the climate in the state where they were found. So yeah, they may have genetic traits that adapt them to that. And of course, diet could have something to do with it. There could be a number of factors that influence how long different populations take to hatch. So number 2, what is the research question for this exercise? Basically, is there a significant difference in incubation times in prairie lizards from Arkansas versus Missouri? That's a real simple research question. It says write a biological hypothesis. You can do if and then, or just if then. But this is, these are sometimes, wordy. It depends on how you want to word it, but this is a biological hypothesis, which is when we explain why what we expect to happen and why. And so an example might be, I don't know, let me do something here. I'm going to try to freeform a hypothesis here. It might be ugly, but I'll work with it with you here. Let's go to a whiteboard. I might say something like, so once again, a biological hypothesis is an explanation, not just what you think will happen, but why. If Missouri lizards are adapted to a cooler climate, I'm not sure on this. I'm not a lizard expert, so I'm just kind of free forming here. They may hatch, they may hatch faster than Arkansas lizards. What I mean by that, what I'm trying to say here is, if you're living in a climate where it's cold, you want to get out of the egg as fast as possible, so that way you can bask in the sun and stuff if the temperature's cooling off. That's what I'm trying to say, but in a real short way here. They may hatch faster than Arkansas lizards. So I'm going to say, so actually, let me clean that up. I need to fix that. To a cooler climate, then they will hatch faster than Arkansas lizards. And I can make that more clear. I could work on that if part, the why I predict this. But that's a very short hypothesis there, but I at least have an explanation. They're adapted to a cooler climate. I could expand on that why I think that, but that's a nice short biological hypothesis. Okay, now this is, once again, that's a biological hypothesis. What would a null hypothesis be? So the null hypothesis has to do with the t-test. What we're going to either accept or reject the null hypothesis. What would the null hypothesis be?
That there's no difference in how fast they hatch.
You can make this longer or briefer. And H1. Now, I kind of predicted directionality in my biological hypothesis, but just for sake of simplicity, let's just say there is a significant difference. We could do a one-tail test and predict that Missouri will be faster, but with the t-test, let's just look for a difference. How about, I can even reword. I can't. It won't let me edit this. I could edit my biological hypothesis to say then there will be a difference. I could do that to make everything match up really nicely. But it won't let me edit my biological hypothesis. If I click on it, it doesn't work. It won't let me edit it. All right. So here was what we have. So what I want you to do for now is I'm gonna break you out into rooms. You can work on it on your own quietly, and then ask for help, and I might hop in and out of the rooms. I want you to try to graph this data, and I want you to put error bars on it, standard error bars, and I want you to do a t-test on this lizard data to see if there is or is not a significant difference in the rate of incubation time, the hatch time, between lizards from Arkansas and Missouri. And remember, this is actual data collected by a UCA professor, so it's kind of cool. I don't even know if it's published yet or not. It probably is by now, but I'm not sure. So I am going to stop this share. I'm going to break you out and I'm going to hop around and check on you. TAs, please help folks as well. And this is, I think, page 3 of the data sheet that I sent you. So let me go in here and go to breakout rooms. That way you don't have to talk to everybody. Oh, Kira has a meeting. Okay. Interesting. Okay, so what I'm going to do now, if I were in person, we'd walk around and I'd just look at all your computers and see what you got. But what I'm going to do right now is, I don't know what the best way to do, the best way to do this is. First of all, I'm going to share my screen. I hope you can see my Excel sheet. Is that a yes? Julia, can you see my Excel sheet? Oh, she's talking to somebody. Hi, whoever Julia's talking to.
Yeah, we can see it.
Okay, thank you. All right, so what do I have to do to start? Where did you start? Where can I start here? Somebody.
Start by finding the averages.
All right, so I'll show you how I do it. I always paste the categories down here first. Actually, I'm going to make my life easier. I'm going to go to insert. Oh, no, never mind. No, I'm not. Let me paste this first. I'm going to paste those same labels down there, and I'm going to give myself an extra column here. That way I can label things. So I put these two labels that are up here down here again. It just makes it easier to graph later. It makes them show up on the x-axis. And now I need to do, like you just said, I need to calculate averages. And how do I do that? Equal sign. What do I type?
Average.
Average. Yay. All right. Open a parenthesis, highlight those numbers. I probably can just hit enter and yeah, you don't have to close the parenthesis. I just do it out of habit. You want to know a shortcut? If these two have the same number of samples, I can copy this and paste it here, and it copies the formula and not the actual value. So you can copy and paste the calculation. It doesn't work if, like, Arkansas had 10 samples and Missouri had 15, then it wouldn't work. But if they have the same number of samples and they're lined up the same way, it'll work just fine. Okay, standard deviation and standard error. Standard deviation, anyone remember the formula? What I would do, if you haven't done this, or if you're new to this or it's been a while, I would go ahead and jot some of this down too. So you have a set of your own instructions to refer back to when you're doing the assignment for today's lab. By the way, that would be a good idea. So, like, if you don't remember how to calculate averages, if you don't know how to calculate standard deviation or how to graph, those sort of notes would be invaluable here. So standard deviation, what's the formula?
ST diva.
ST diva, standard deviation. You open a parenthesis once again, you highlight the data, and either close the parenthesis and hit enter, or just hit enter and it'll copy it. It'll do it. And once again, you could copy and paste this into this cell and it would do it for you. I'm going to go ahead and do it again though. Equals ST diva, open a parenthesis, highlight Missouri numbers instead of Arkansas numbers, close my parentheses and hit enter. Lastly, standard error. This one's a little bit different. We do equal sign. What do I click? For standard error, I have to divide standard deviation by the square root of samples. So what I put equals, and then what do I do?
You select your standard deviation.
Okay, select my standard deviation. In this case, that's that one right there, that 1.03 or whatever, divided by square root is like squirt, SQRT. And I open a parenthesis again. I got to tell it the square root of what? And that's the square root of the number of samples. So how many samples did we have in Arkansas?
10.
10? Boom. All right, there's my standard error. Once again, I can do the same thing for Missouri. Equals, click on standard deviation, divided by the square root, and it's the same number of samples, so the square root of 10. And so that's what I got for those. All right, now how do I graph this data? Actually, before I even graph it, let's do the t-test. Equals, what do I type now? Something.
T-test.
All right, t-test. Okay. Open a parenthesis, and it tells me what to do here. Array 1, array 2, tails, and type. So array 1 is that. The Arkansas values. It's the raw numbers, not the average or anything, just the raw numbers, comma, Missouri, numbers, comma. I'm going to do a two-tailed test. So number 2, comma, and then the number 3, unequal variance. And I hit enter and I get a value. Now, that number is a lot different than the one we saw in the last t-test. What in the heck is that number? What does that mean? Is it a big? Oh, go
It
ahead.
has scientific notation, so it's super, super small.
It's super, super small. Yeah, it's like 4.02 times 10 to the negative, a lot, 5 or whatever there. So it's a very small number. It's way less than 0.05. So first of all, are these two samples significantly different based on that calculation, that p-value? Is that less than 0.05 or greater than 0.05?
Less than. So
It's way less than.
It's significantly different.
Okay, so they are significantly different. Now what we need to do is we need to put in a graph. So I'm going to highlight those two values, and on my version of Excel, I go to insert, and I'm going to, you could insert all kinds of different graphs. Like I could click the wrong kind of graph. That graph would make no damn sense, right? Like, okay, what do I do with that? That doesn't look good, but I want to go to insert, and I want to go to column graph. And there I have a nice column graph. I'm going to delete my chart title. I'm going to delete these sideways bars to clean it up a little bit. Okay. How do I add error bars? Someone walk me through this process.
Okay, you go to add chart element.
Okay, which I can do up here or this plus sign down here. Let me try it up here. Let me just see. If I go up here, it's right here, error bars. Okay, what do I do then?
You go to more error bar options,
More options. Okay.
and then
And-
you click on the
And notice
little-
mine is showing the wrong menu here. It's on the paint bucket. I need to go to the graph. Sorry. Okay, go ahead.
you go on the little 3 things. Then you hit custom.
Okay.
Specify value,
Okay.
and then you click that little thingy right there,
Uh-huh.
and then you highlight the SE for both of them. the
For
standard
both.
error For both.
Exactly.
Then you
Okay.
hit, and then you do it again in the negative.
And do it again. All right, same two numbers. And then I click OK. All right, so there's our data. And now I need to, once again, I need to add in axis titles, and I can call this location. And I could call this one incubation time. And that'll be days as the unit in parentheses there. All right, we did it. Now, we're not done though. This is just a graph. If we want to make a figure, we have to write a caption yet. And so if you haven't done that before, I'm going to copy and paste my graph into a Word file. And let me make sure I'm sharing that with you. Share. Okay, so I copied and pasted this into a Word file. I can make it a little bigger if I want. It's always nice to be able to see what you're doing. And then click underneath of it. And this is our first figure that we're making. I mean, normally we only make one figure for an assignment, for example. So we would just call it figure 1, period. By the way, there's an example of a typical Bio 2 column graph figure. Please hold. I gotta find the page number. If you look on page 109 and 110 in your lab manual, there are example figures and captions. There's examples of what to put in a figure caption. There's a list of things. So there's also an example, and then there's some examples of how to calculate change and things when we're writing about data. So that's Appendix A, starting on page 109, and it goes through page 111. That's handy information for you there. So if we look at the instructions for what goes into a caption on page 109, we first of all remember that figures, you're always going to turn in figures, not just graphs. So a figure is a graph with a caption. And so if we go down to step 1 under figure captions, they should be brief and contain enough detail to explain the figure. We're not going to really include conclusions of the data from the data, but just what happened. So the first thing we do is we put the word figure 1, period, and then we're going to write our title for the graph. So does anyone want to venture to give me some kind of title for this? Like, what could we call this thing? Like, would this be a good title? Hold on, let me zoom in a little bit. Is that a good title? Why or why not?
It doesn't really explain anything that happened.
Right. It's really crude, really short, not very descriptive. We want a nice descriptive title. So we could say, okay, incubation times of, and it says in the description of what kind of things go into a figure caption here, that we ought to have the name of the species if possible, things like that. Actually, I have to go back to the lab manual because I forget the scientific name of this species of lizard off the top of my head. I bet I have some. If I have any Gifford students in the room, they'll know it off the top of their head, but I do not. Here we go. So if I go back to the data table, I'll say incubation times of prairie lizards, and then I'm gonna misspell prairie like I always do, and then in parentheses I'm gonna put what the scientific name is. And I'm gonna italicize that so that we haven't talked about this, but we always italicize our scientific names. All right, so that's an example of a title. Then we have a second sentence, and that's going to be a description of what happened. So I could say, Arkansas lizards had a X% longer incubation time than those from Missouri. All right, so we have to calculate that number. If you took Bio 1 here, you've done a lot of percent changes already. If you don't know how to do percent change, or percent difference in this case. The way you calculate that is shown on page 111. Y2 minus Y1 over Y1. So depending on how you set this number up, you're going to get a positive value or a negative value. So I'm saying that Arkansas lizards had a longer incubation time. So that's going to be a positive value. So I have to set this equation up the right way so I get a positive difference. So Arkansas has a higher number. So that's going to be Y2. So Y2 minus Y1 over Y1. So Y2, what was the average? Let me just go to Excel real quick. We'll just do it in Excel. Let me share that screen. Do you see Excel or do you still see my my Word file? Okay, still see the Word file. Okay. New share. We're gonna share. Let me just share the screen, then you should be able to see it. Hopefully you see it now. So I'm going to just calculate it here to make it easy since we're all on computers. Y2 is whatever the average for Arkansas was, 55.8. Actually, let me put that in parentheses. 55.8 minus whatever the average for Missouri was. That was 53.4 divided by, what is it, Y1, which is 55.8. And then, oh, I forgot to do, and then you multiply that all by 100 to get percent equals that times 100. Oh, that did not work at all. I screwed that up. There we go. Now, if I did this right, all right, so what I did is I took this number minus this number divided by this number, and then I times it by 100. That's all I did there. It's not really that complicated. And if you ever need help with that, you can Google percent change calculators on the internet, and you can use those online too. It'll be like, what's the first number? What's the second number? And it'll spit out a percent change. Now, you will set that equation up differently. Like if you said Missouri had X percent lower incubation times than Arkansas, you'd have to reverse Y1 and Y2 because that would be a negative value, which shows that there's one lower than the other. It only makes intuitive sense when you play with it a little bit. There's no way for me to just say it out loud and it all to click. Just take those two averages and look at the different ways you could calculate the change, and that'll help you to make sense of that. Okay, now there's a couple things we need to do yet. We're not done. We are going to, if we look at that example caption, or I'll just do it here, I'm going to open a parenthesis at the end of the sentence here. I got to tell the reader some stuff. First of all, I have to tell the reader that these columns are averages. Does anyone remember how you do that? What's the word? What's the fancier way to say average? Mean. And then I have to tell them that these are standard error bars, because those could be standard deviation bars. Those could be range bars. They could be all kinds of things. I have to tell them what those bars are. So these are means or averages, and these are standard error bars. The way to do that is mean. I have to insert a symbol that says plus and minus. Now, this is often the hardest part of the whole thing when you're first starting Bio 1 or Bio 2 is finding this damn symbol, because if you don't use it, you got to dig for it the first time. And then once you find it and use it, it shows up in your recently used symbols. To do that, you go to insert at the top of the screen in Word, and then you go to symbol. And then you have to find it. You might have to go to more symbols and hunt for it, but it looks like this. It's a plus sign with a minus sign under it. And I'm going to stick that right there. I'm going to type in SE. Actually, let me just finish this caption and then you can hunt for that symbol. The other thing we need to do, I'm going to put a comma and I'm going to say N equals 10. And you could say n equals 10 for all samples, or just n equals 10, since n is 10 for Arkansas and for Missouri. If the sample size was different for the two different states, you'd have to make that clear in the caption, but it's the same. I'll just say for all samples, or for all how about all locations? Or I guess I could just say both locations would make more sense. All right, so that's everything I need in my caption. So I have a title. I quantified. It's important that you quantify the difference between these two data points. How many times different were there? How many % different were there? What was the fold difference between them? So there's information on ways of doing that. And if you need extra help with that, you can stick around at the end of the lab, and I can help you work through that. Or you can come to office hours, but you quantify the difference between the two. You don't want to just say Arkansas had 3 days more incubation time than Missouri or whatever. You want to give it a quantification. How many times different, how many % different, that sort of thing. And then at the very end of the caption, we have this statement in parentheses, which tells the reader that those bars are averages. I'm sorry, the columns are averages, the error bars are standard error, and how many samples were used for each of those columns. How many samples did you collect to get those averages? That's what that n equals 10 tells them, the sample size for each of those columns. So that's what goes into a caption. Okay. So you should, hopefully you have this exact thing on your computer now, having done all this, but if you don't, stick around and I will work with you to help you. But meanwhile, those of you who are all caught up and are feeling confident about it, on Blackboard, let me see if I can do this. You're seeing my web browser right now, aren't you?
Correct.
Okay. Please hold. All right, let me switch to student view. By the way, Science Wednesday is this week. Remember, you need to attend one of those during the semester and write a little summary of what you've learned and relate it to class material, just a half-page little summary of what happened and what you learned. I have instructions on it posted online. There's about 4 of them throughout the year, the semester, and the first one is this Wednesday, and there's the Zoom link for it right there. Okay, if you go to lab within Blackboard, oh, apparently I screwed this up. Never mind, I'm a liar. Let me exit. I must need to turn it on. I must not have released it to you yet. Yeah, here it is. Let me make it available. Make available. There you go. Now it should be available. I'm going to look at it as a student again. Okay, so in here, the research question. So I have a file here that you can download. And the research question is there a significant difference in the average number of daily cases of COVID in January 2021 versus January 2022? The 2021 would have likely been caused by the Delta variant, which is a different genetic strain. So evolution's at play here, versus the Omicron variant, which is around now. And I have your null and alternative hypothesis already written for you. I want you to create a figure, which is a graph and caption of the attached data, run a t-test. And I want you to, so remember, it's a figure. You're going to have your graph, you're going to have your caption. And then I just want a 1 or 2 sentence conclusion answering the research question. Were they different? Were they not different? And what I want you to do is you'll have a 2-page document you're working on. You'll have your graph and caption on one page. And I want you to paste your Excel sheet after you've done all your calculations on the second page of that Word document. Then save it all as a PDF before you turn it in. That way, if you make an error, I can at least kind of see what you did, so you can get partial credit if you calculate something wrong. So you'll have your graph and caption and your 1 or 2 sentence conclusion on one page, and then your Excel sheet copied and pasted on the second page of that Word document. And what you'll do is you'll download this file here. It's called COVID variants, and it's actual data. from Arkansas for the first 15 or so days of 2021 and 2022. Okay, so there's 15 days worth of data here, and it's the number of new daily cases for each of those days. So it's going to graph just like the lizard data did, only a different sample size. And you're going to do the same thing with a graph and caption, and you're going to upload it before you come to lab a week from today. It's due before you walk into lab. There's no due date on Blackboard. I can't give you a due date and give tomorrow's lab a separate due date. So you just have to know it's due before you come to lab. So you might set a reminder in your phone or whatever. It's due before you come to lab next week. And that will be your assignment to show me you know how to work with this kind of data. Okay. So I'm going to stick around. That was lab 1. Next week is lab 2. Let me bring the PowerPoint back up before I forget. So just to wrap up some things here. This was just a hands-on, how do we deal with data kind of lab. That way in the future, when we collect real data, you'll know what to do with it. So graphing allows us to visually see what we've done with our data. And if you have data that's continuous on both axes, so that means like lots of numbers on both axes, that would be a scatter plot. But if you have discrete data on one axis, that means like a category, that would be a column graph, like lizards from Arkansas versus lizards from Missouri. If it was just lizards and you had a different or a range of temperatures on one axis and incubation times on the other, that might be a scatter plot that you would graph it with. But the assignment you're doing for me for next week, that's 2021 versus 2022. That's a column graph, just like you did with the lizard data. Okay. It'll be 2 columns. If you graph it and you get a bunch of different, one of the biggest errors students make is they turn in a graph that looks like this one. That's because they forgot to average their numbers together. It looks like a cityscape with a whole bunch of columns if you don't average your numbers together. You have to average 2021 together, average 2022 together, get the averages and graph those. So that's one of the errors that students often make. All right, so just to remind you then, what goes into a caption, I do have a slide of this. You have a figure number, you have a title, and then you're going to have a description of the change, and you're going to have some indication in that caption of the sample size, the mean and plus and minus SE. And the one thing I didn't do, I actually forgot to do, I'm glad I brought this up, is put the p-value in there as well. So I'm glad I brought this up. I would have forgotten to show you that. I would be a terrible person. All right, so let me open this back up. Now for this example, P was less than 0.05 and it was a tiny little number. So I'm just going to shorthand this and say P equals less than 0.05 comma, and put that in that caption as well. So, oops, I have 2 commas there. Now, if that p-value was not a tiny number with an exponent, I would ask that you type the number in. P equals 0.0261 or whatever. But it was a tiny crazy number, so I did P equals less than 0.05 in this case. But the good news is, if your number is that small for your homework, you're going to turn in your Excel sheet, so I'll be able to double check that your p-value was correct, so I can see that. Even but if P is something you can easily type, go ahead and put the actual value here, just out to like 2 significant figures or so. You don't have to put a really long number there. Okay, so I'm glad I didn't forget to show you that. Okay, and label your axes. And I can put this PowerPoint also up on Blackboard at the end of the lab, so you have access to it. So before we go, let's say you had this graph. What could you do to this graph to make it better? What is it missing? What kind of things does it need?
Add error bars.
Okay, error bars are definitely missing. So control and caffeine, they don't have error bars. Thank you. What else could it use?
You could add like some quantitative values into the figure 1 description.
Yeah, so we could say students who drank caffeine had a whatever % higher pulse rate. So to calculate that, we could call this Y1 and this Y2 because it's higher, right? So we would take this average, this value, y2 minus y1 over y1, and we could get the value we're looking for. But students who drank caffeine had a whatever percent higher pulse rate. What other stuff is missing on here?
The mean.
We don't
and
have
the
the
p-value.
Yeah, the mean and the p-value and all of that. We don't have all that stuff that we did a second ago. Now I want to point out once again, students sometimes get confused on this. You actually literally type the word mean. Sometimes students think you have to type what the mean was there. No, I literally want the word mean. That tells the reader that these columns are averages. That lets me know that those columns in the column graph are averages. So mean tells the reader, hey, those are averages. And then plus and minus SE tells me, oh, this is one standard error above and one below that average. This little description here is very important so I can understand how to make sense of what this graph is, what you did to make that graph. That's why we do those things. Okay, so you're going to make a figure of the assigned data, do the work on your own. If you get lost, you can rewatch this video. There are videos on the lab website, and I have office hours. My office hours are Tuesday and Thursday, 10:50 to 12:05, Wednesday, 1 to 2:50. And they're on Zoom, and you can get to it through Blackboard if you go to the Zoom link for me. If you want to, I will have info sometime soon about SI if you want to go to one of those sessions. We don't have it set up just yet. Next week, unless I have to change plans because of COVID or whatever, the plan is that we'll be doing lab 1 in the manual. And if you happen to live near a lake or pond or creek, go ahead and bring in a little water sample. We can see if there's anything alive in there. And the best way to do that is not just to get a nice clear cup of water, but get some gunk and leaves and muck from the bottom, because that's where critters are hiding and living and feeding, is in the stuff that's in the water. They're not just swimming in the clear water. They're hiding in amongst the habitat within that water sample. So I'll try to bring some samples from my pond. If it looks like everything's dead, like if it gets too cold, we might switch labs and do this lab when it's a little warmer and there's more life out there, because sometimes it's really boring if everything's dead. But we usually can find something early in the spring. It's always better to closer to summer, but I want you to learn to use microscopes early on if we can do that. So that's the plan for next week. And you can watch the videos. There might be a little quiz next week when you come in over some of the stuff we talked about today, or over the pre-lab reading for next week. Just make sure you can answer like those pre-lab questions at the beginning of the lab, and you'll know you're good to go. So that's it for me. I'm going to hang out and help anybody who has questions. Please don't be shy. Don't hesitate to ask for help. If you are like, oh my God, I have no idea what the hell just happened, that's what I'm here for. I'll stick around. Otherwise, I'll see you in lecture tomorrow in person. Hopefully that's the plan, right? Tomorrow morning.
Hi,
Hey.
I do have a question.
Sure, no problem.
I'm on a brand new computer. I'm on
Uh-huh.
a Mac, and I've never had a Mac before, so
Oh,
I'm
sweet.
learning a lot right now. I did not catch how to add the standard error bars to the graph.
Okay, let me stop the recording because you probably don't want this part